Imagine.
Good afternoon, everyone, and welcome to our annual monthly Firefighter Cancer Initiative monthly seminar series. So today we are really excited to have Dr. Alberto Caban-Martinez and Dr. Natalia Suarez from FIU present today. So I know people are still logging in. I'm just going to give everyone a couple minutes, and then I will introduce to you our speakers and their topic. All right, so I'm going to go ahead and get started. So today we're really lucky to hear some results from the evaluation of a non-invasive method to measure per polyfluoroalkyl substances exposure in firefighters. So I know this is a hot topic for our firefighters who are always curious about their gear and want to know about PFAS exposure in the fire service. So today we have Dr. Natalia Suarez from the Department of Chemistry at Florida International University, which has been a great collaborator and partner with us here at University of Miami and the Firefighter Cancer Initiative. And co-presenting today is Dr. Alberto Caban-Martinez, that many of you know. He is an associate professor here at the University of Miami in the Department of Public Health Sciences and serves as the Deputy Director of the Firefighter Cancer Initiative. So thank you both for being here and presenting your work. So without further ado, I hand it off to you both.
Wonderful. Thank you, Dr. Sali, for the warm introduction, and good afternoon, everybody, for our monthly Firefighter Cancer Initiative seminar series. I'm really delighted and humbled to be presenting with my co-presenter today, Dr. Natalia Suarez from FIU. This journey began almost like 2-3 years ago when we started having conversations about PFAS, and particularly in the space of our with our work here at Sylvester on exposures among the firefighters. Dr. Suarez, as you'll learn, is an international expert in this space of PFAS, and we're really fortunate to have teamed up with her on a project to explore a different method of how we can potentially evaluate PFAS on the gear of firefighters. And so this is sort of a pilot project that we're really excited about. So we've divided our presentation that I'll give some background information on the topic as well as the methods for this particular project, and then I'll turn it over to my colleague, Dr. Suarez, who will go over the methods for PFAS analysis and the findings, which are really exciting to see if the use of a non-invasive textile is good at sequestering and identifying PFAS. So we would welcome your questions. We ask that you post them in the chat, and then towards the end, we'll open up the webinar for questions from those in the audience, as well as entertain those that are in the chat. Okay, so let me begin first by just quickly acknowledging the funding for this research project. So both Dr. Suarez and I were funded by the state of Florida appropriation to the Sylvester Comprehensive Cancer Center for this Firefighter Cancer Initiative project. So we're thankful to the state for those fundings, as well as some support from the National Cancer Institute as a program grant to the Cancer Center. We want to remind everybody that the content that we're presenting to you today is those of us, Natalia and I, but does not necessarily represent the views of the state of Florida, the NIH, FIU, or University of Miami. This is a research project we've taken on ourselves. All right, so beginning with what is PFAS. So PFAS is a really large and broad family of chemicals. These perfluoroalkyl alkylated substances have been around for some time, and they have another pseudonym as they go by forever chemicals. And they're forever chemicals because they tend to have a long life in the environment and the human body after, once you've been exposed. In fact, one of the types of PFAS, PFOA, is said to have a physiological half-life of about 2.7 years. And similarly, PFAS, another type of alkylated substance, alcohol substance has about 3.4 years, and PFAS itself up to 5 years. And so these half-lives really vary, and it has to do a lot with the structure of the chemical. So for example, on the right, we have an illustration of a particular type of PFOA, which is one of many types of PFAS, and you can see it's a carbon fluorine bond. They're very strong in nature, so to break them, it takes a lot of energy. So that's why we say that they are forever chemicals. They last for a very, very long time. Health studies that have been emerging in the literature have started to link PFAS in serum and in blood, both in animal studies and now some human studies, looking at how it's tied to immune function, so how the immune system can respond and react to it, as well as some cancer outcomes. So of course, we'd be very interested in this health topic for our first responders and other community members of how PFAS potentially impacts health. So where do we find PFAS? Where is it in the environment? So this wheel is a reminder of like so many different point sources where PFAS is there. PFAS is a very good water repellent. Its bonds are strong enough that it pushes water away. So we see these PFAS chemicals used, for example, in paints, in shampoos that are used commercially at home. So for example, non-stick cookware, contemporary commercials on TV might tell you, well, this new cooking set is, PFOA free, that doesn't have that PFAS on it, right? And so before, we used to actually coat cookware to prevent some of that. You might see it in some of the materials used in black rooms for processing photography, in fast food packaging and pesticides, and more importantly, firefighting foam, as well as gear, which we'll talk about in a second. And so for firefighters, we suspect and have some suggestions that PFAS can be found in the safety turnout gear. So the same gear that's used to protect our firefighters might also be lined with some of this PFAS, particularly the inner liner. So firefighter gear has different layers, and one of the layers is a water barrier or moisture barrier. And Dr. Graham Peasley, who recently published a paper showing that he was able to find some of that fluorine present in that turnout gear, suggests the presence of it there, as well as the foams that are used by first responders to suppress particular types of fires. So we see that these aqueous foam forming foams contain some PFAS, family of PFAS in there. So now the question becomes, does your gear expose you to PFAS, right? And how do we determine what type and how much of the PFAS is in that gear? So the concerns with PFAS in the gear arrives primarily from two main sources. One is the natural aging and breakdown of the gear. So as firefighters use their gear, there's obviously some more natural wear and tear of the PFAS, and that aging process affects the textile, which in turn may leach some of the PFAS chemicals out of the textile and potentially be absorbed by the firefighter through the skin, right? And this through the water repellent liners. The other potential sort of area of concern or point source, as we mentioned, is in the use of AFFF, right? So if a firefighter, let's say, is cross-contaminated with some of the foam in the process of doing some of that fire suppression, some of that foam remains on the turnout gear. And so when they're exposed to sort of some of that high heat, some of that fluorinated material can seep out. So it becomes a little tricky, right, at how we find, what PFAS is on there, and how do we measure it, and what types are present. So we do think about some restrictions that are challenging to sampling PFAS in gear. So the number one is do no harm, right? We definitely don't want to cut up the gear of a firefighter who's working because they need their gear and they can't have a hole in their gear, right? So, how do we sample gear that doesn't destruct gear that is very expensive and that firefighters are actively using? The other sort of restriction is that turnout gear has to be immediately usable, right, by the firefighter. They can't really have downtime, so sampling can become tricky in terms of when and how to sample, given when the gear is in use. It may be needed at any moment's notice, and so stopping it when it's wet or treated with other flammable solvents is not possible. It makes it very difficult. The other sort of like lens that we think about sampling the gear is using sort of like everyday items that may be contaminated with PFAS itself. So for example, latex gloves, prefabricated swabs, certain tubings and the septa of those tubings can be manufactured with PFAS. And so, if the gear touches it, is there cross contamination of the PFAS from those extraneous materials on the gear where we're trying to sample? And so as scientists, we have to be very careful in the procedures of how we sample the gear for PFAS. So like a consideration is the glass vials that we traditionally use, say for wristbands to measure pHs, really can't be used, as those glass vials might absorb some of the PFAS and artificially manipulate the type or concentration of PFAS that we want to see in the gear. So for this project, we're going to share with you sort of the methods of how we went out to the field. We worked with South Florida Fire Department, and as always, we're very thankful to our firefighters for the opportunity to collect these samples and work together. We used a special textile, so we used a textile fabric that's made out of polypropylene in order to use that textile to swab against the firefighter turnout gear. And this is a special textile that's helpful in trying to avoid PFAS contamination. So we cut out swatches that are 2 inches by 2 inches in shape with clean stainless steel rotary cutters to make sure that they were not PFAS contaminated. And then we place those clean cut swatches of polypropylene into LCMS grade water, like liquid chromatography mass spec grade water, and about 10 mLs to suspend it there. Then the fabric was handled with very clean forceps, as you can imagine, and wearing nitrile gloves in order to avoid cross-contamination of any PFAS in the preparation of the textiles and then the transport between the laboratory at the university and the fire station. So what did we swab? So we had several huddles on the best approach of how to swab. And again, we're very thankful to Dr. Suarez and other colleagues to provide input on how to do this best. But we selected 12 sets of turnout gear from a local South Florida Fire Department. And we used the moistened polypropylene fabric swatches to be able to abstract the PFAS from the gear. So the process is to use circular motions. You take the textile and for 10 seconds, you swab it around a particular area of the gear. We try to stay roughly uniform in the number of swabs. So usually it was one particular team member that was doing it so that there would be consistency in the abstraction. Nitrile gloves were worn and exchanged after each swab in order to avoid cross-contamination between that swab and the gear that was just touched. So what did we swab? What areas of the gear were used? So the SCBA masks, we did one on the inside and one on the outside. We also swabbed the right and the left shoulders of the turnout gear. We swabbed the right and the left shins. So that's the area between the knee and the ankle, sort of the pants of the turnout gear. So we had a total of 6 swabs per set of turnout gear. We also had some controls to make sure that along the way, as we're collecting this samples, as well as in the lab, we were seeing what we're seeing. So we had negative controls, and these were prepared polypropylene swabs in a vial that never left the vehicle during sample. We had triple blanks. So we prepared the same swabs in a vial that was not open during sampling, but was transported to the actual fire station for turnout gear testing. And we also had positive controls, and these were prepared polypropylene swabs that were dipped into an undiluted AFFF foam to be able to control for those samples. So now I'd like to turn it over to my colleague, Dr. Suarez, who's much smarter than me and can explain our methodology for testing the type and quantity and the findings. Over to you, Dr. Suarez.
Thank you so much, Dr. Kevin. Can you all hear me well?
Loud and clear, yes.
Okay, cool then. Yeah, so as was mentioned, now I'm going to be talking a little bit more and starting first, once we receive those samples, from the whole team from UM, they went on the field and collected them. So the first thing we had to do was to cut this polypropylene fabrics in small pieces, because otherwise it would be a hard task to try to extract them. So we put them, I think I can even I probably cannot put, oh no, yeah. I was trying to see if I can show it each of the stuff, but I'm just going to talk it about, yeah. So then we have a centrifuge tube where we put those small pieces of the fabrics. We added the appropriated label internal standards, and added methanol as extraction solvent. We put it in ultrasonic extraction bath, as you can see here in this figure. We left it for 30 minutes, and after extracting it, all the PFAS, we got it absorbed into the fabric. We centrifuged them. We took it this supernatant, and we evaporated. And of course, when you're adding methanol to the fabric, you can have actually some pieces of fabric that can come out. So we actually had, before putting in a LC-MS instrument, we had to filter them. And I wanted to acknowledge, you see here is a picture for the undergrad student that was helping it. He did most of the sample preparation and analysis as well. You can put next. So here you see is a picture from the instrument in my lab. So we are using an Agilent 6470 LC-MS, so it's a tandem mass pack, which is equipped with an electron spray ionization source. You can see, from different from the majority of other instruments, this one has been modified. So all the PFAS, all the tubings has been modified to be PFAS tubing it. And on top of this, we also use a delay column. And the reason is a delay column is a column that goes between the mixer and the injection port. So to make sure that even if we have, as Dr. Caban Martinez before mentioned, you can actually have these compounds everywhere. Even sometimes methanol, you can have some sort of contamination. So just wanted to make sure that, if we have anything, this would not, we're going to be shifted away from our compounds, from chromatogram from our real bit that could be coming from a sample. We use an analytical column, a PFP. Don't be scared. This is doesn't mean that it's a PFAS column. This is a C18 column. So it's a normal use for reverse phase chromatography, but it has peripheral pentafluorphenyl. These are some of ligands, and they're actually very helpful to increase selectivity and separation from allogeneic compounds. So that's one of the reasons. So trying to make a better separation from PFAS. Mobile phase, we use ammonium formate as a buffer and methanol for separation of those compounds. Injection volume, as was mentioned before, we had some samples. There were actually echo samples. And those samples normally we can directly inject in our instrument, and we do a higher volume injection for 100 microliters. But from the extracts that I mentioned before that are done in methanol, they are 1 milliliter extracts, and normally we only use injection volume of 20 microliters. We do the type of acquisition of this method is multiple reaction monitoring method. We normally try to see 2 different transitions from PFAS. It might be too much analytical talk for here, but just making sure that we wanted to make sure that we're seeing what it is. And so one of the major issues in analytical chemistry is that, for example, for PFAS, some of those compounds actually, don't have 2 transitions. So is sometimes you're going to have only 1 transition, which makes it harder sometimes identification. And I'm going to talk a little bit more about this when I talk to quality assurance, quality control. So after running it, all those data was then processed with our Agilent software, Mass Hunter. Next. So I just wanted to show, we analyzed a total of 30 PFAS, and those included both some of the long chain PFAS, legacy PFAS, and from different classes, as you can see here, PFCAs and PFOSAs, PFSAs, and PFOSSA as well. Can you put it just the next slide? Next slide. Yeah, so as well as some short chain PFAS, which show some of the same classes that we saw before, also include some of the fluorinated ethers, some that people have already known, like Genax and Adona. Next. So just talking about some of the quality control, quality assurance that we took it in the lab. We have made, a calibration curve that was 9 points. It was slightly different in terms of lowest concentration in water. It was from 2 to 1,000 part per trillion. And then for methanol was normally from 5 to 1,000. It was always run in the beginning and the end of the batch to make sure that, all the conditions are being maintained. We normally see a very good linearity. Besides all the negative controls that was mentioned before for the water, methanol, the field trips, the positive controls, we also have in the lab procedure blanks, spike blanks. So spike blanks is when you add the set of native standards to the sample, to the blank sample. We have a continuing calibration verification, and we also had what we call an ICV, is initial calibration verification. So this ICV is normally, is a secondary standard, so it's bought from a different source. So to make sure that, your measurements are accurate. And again, all of this type of samples were included in every batch of 20 to 30 samples. And, normally the CCV was included, every 10 samples, just to make sure that, the performance of the instrument was maintained through the run. So how do we say that we have identified and have a quantifiable PFAS in a sample? There are some criteria. Normally, they have to have a rotation time that doesn't shift more. It has to shift the maximum of 0.2 minutes from the corresponding isotopic label standard. This is like the majority we have. In some cases, we don't have it available commercially. So in this case, you would compare, the rotation time to the rotation time that we saw in the standards, right? And compare with the sample with the standards. We also had to check the presence of the confirmation peak, especially, most of the time we had 2 transitions. This is, particularly to make sure like you're really seeing what you're supposed to be seeing. Some contaminants might have like, a similar mass than PFAS. Especially, not in this type of sample, but especially when you work with biological samples, there are some fatty acids that have like, similar masses of PFAS. And then the other criteria is that the method detection limit, so all the concentration has to be above the method detection limits, right, within the calibration curve. And the signal to noise, it has to be above 3. So just to make sure that we're not really integrating any thing that is noise from the chromatography. Next. So the method detection limit, the way that we have established the method detection limit is that concentration was, the lowest concentration from the calibration curve that had a signal to noise above 3, and that it had, no more than 30% variation compared to the expected true value. So as you can see here, for the majority of the PFAS, the lowest calibration point, for the water, as I mentioned before, was 2. The lowest for methanol extracts was 5. So we can see that the majority of the compounds that could be seen at a very low concentration. There were some exceptions, especially some of the longer chains in the end, that in methanol they had, a little bit more higher method detection limit. So the signal to noise was not sufficient to distinguish from the background. Next. But I think like that's probably what everybody was hoping to. So now we're gonna just talking about some of the results. So here is just a table looking at some of the results from the negative controls. I just wanted to show you that, like the majority of them was below our method detection limit, but we did have like, some PFAS that it was still quantified in the blanks, the controls. And this is important. That's why one of the reasons we do need controls, because, these compounds, like is mentioned before, is everywhere, and we have to be able to account for anything that could be coming at. It could be coming from methanol, it could be coming from handling the sample, it could be coming from even like, filtering the sample, any step. So we have to make sure that we are counting this. Next. So this here, it shows you is, the results from the control sample. So this is actually very interesting. And I wanted to also acknowledge, you know, the postdoc from my lab. I think she's also watching the talk, you know, Dr. Leila Lemos, you know, has been the one, you know, that has great in statistics. So, and R software. So she was able to make those really nice graphs. So as you can see here in this graph, you know, we saw that, you know, the highest concentration, you know, was from 6,2 FTS. So probably those foams are some more of the newer foams, not the ones or PFOS. We also saw like some higher amount of PFHXA. So like these two compounds are some of the, they're known to be more in those newer foams. And, you know, we also saw some of the other compounds like, you know, PFBA is not really known to be a constituent of the foam. So, I mean, I just wanted to point out that, you know, some of the constituent of these foams, you know, some precursors, they can actually, even though like they might not really have in their constitution, like PFOS and PFOA, they actually might degrade, you know, so most of the precursors, they can actually degrade to this perfluoroalkyl acids, you know, such as PFOA and PFOS, and even leading to the shorter ones. Next, just to show the, yeah, I just wanted to show, like, you know, comparing these two type of foams that were the two positive controls, you know, we can see that, you know, the composition of total PFAS was very similar. And you can see by the concentrations here that they are very high, so in the ppm ranges. Next. So here we see how we can the distribution of PFAS in the turnout gears, right? I tried to put a box plot. I don't know if you guys are seeing some. For me, kind of shows a little bit small, but hopefully you guys are able to see a little bigger. You can see that, you know, we see a different types of PFAS here. But what I wanted to point out, can you just click enter again, like just to show, yeah, okay. So I wanted to point out, you see like the 6,2 FTS, where we saw that the highest concentration in the foam is also the one that has a high concentration in the gears. And I'm here, by the way, I'm gonna have some other graphs, but here is showing you the distribution for each of PFAS and the different gears, like, you know, that was mentioned before. We did an average for the shoulder and chin because it was collected from two different parts, and then we have the information from outside and inside scuba. So we can see here that, you know, the highest concentration was for 6,2 FTS, and we see also here higher concentration from PFHXA. And we also see, like you see the same way, PFBA here, you know, and then there are some other one, PFU and DA compounds that were actually found in some higher concentration, even PFOA. Next. So how does this total PFAS, you know, look like now among the different participants in the different types of gears? So in this graph here, you know, what I was trying to see is, okay, so what are, which gear is actually we having it, you know, the high exposure to PFAS? So we're trying to answer that. So we look at, we can see right away that, you know, that PFAS distribution is a little bit different, right, in the different type of gears, right, and the different also participants as well. So it varies in both. So if we just put next, yes. So this graph here kind of showed us the average of the total PFAS. So we see like more details in the other graph among the participants, and then here we have, you know, in case you guys cannot see it, the yellow here is the shoulder, the gray is shin, and then green is the outside scuba, and the purple is inside scuba. So basically, you know, here what we see it is that, you know, the concentrations. Sorry, did something silly here. So we see that, you know, the concentrations in both shin and in shoulder were significantly higher than the concentration in the scuba, either outside and inside. And interesting, you see that outside is a little bit higher, even though it's not significant difference, but outside shows a little bit higher than inside. Next. So here again, is that it's exposure the same among all the participants. So I put it in this, I tried to put like different types of graphs because, it kind of give you a different perspective from the results. So this one is a heat map. So we can see here, right, like total PFAS, like the highest amount is like, it has more intense, it's kind of red, and then get orange, yellow, and then the lowest amount is blue or green. And you can see in this graph, right, like especially participant one, and I think like from before one, you can see that it has like a lot higher exposure. At least like they had a higher concentration than gears. And this was interesting. If you look at this graph, it looks like at least from 1 to 6, the exposure is a little bit higher compared to, for example, from 7 to 12, This could be for, many reasons. It can be like the type of activity they're conducting, or a duration of it, or even like, if these gears have been previously washed or not. So next. So I think like I just wanted to get some conclusions, and I think like some of what we envision to be some of the next steps. I think like I hope at least that I have showed to you all that we have actually been able to have a very non-invasive and also easy approach for the assessment of PFAS, levels of composition in turnout gears. We have seen like, extremely high levels of PFAS, and the highest amount was actually for the 6.2 FTS that was detected in the shin, was 282,000 nanograms per gram. This is corrected by the amount of tissue. The levels were significantly higher in both shin and shoulder compared to the inside and outside of the scuba. So we did some statistical tests, Kruskal-Wallis test, so a non-parametric one because the data was not normal distributed, with a post hoc test. And PFAS were also significant among the different participants. As I mentioned before, they could be maybe related to the occupational history or activities performance or duration or even frequency of washing of the gears. And, next, what we are actually looking at to do it is to try to understand if we can use this type of methodology to predict amount of PFAS exposure in blood serum, so in blood from the participants. So I think like Dr. Caban-Martinez also have data on PFAS in serum. So we're going to be trying to see if we can try to find some correlation between, levels in gears and levels in the blood. Next. And I think lastly, I just probably missing some people here, but, I wanted to acknowledge, some of my postdoc, Leila and Denny. Denny is the one that always makes sure that the instrument was working properly. The undergrad student that was involved in this research, Kristen, and also all the collaborators, all the team, and I probably forgot, I just put here over to Natasha, but I remember there was Paola as well, if I'm not, and, and also, I know that it was already mentioned, but, we are also appreciated the funding from the University of Miami with the state of Florida from the Firefighter Cancer Initiative.
Awesome. Thank you so much, Dr. Suarez. So this is a point where we'd like to just entertain questions from the audience. So if you would be as kind to type in your questions into the chat room, and we'll take a stab at sort of addressing them. So we do have the first question, and happy to answer it either way, Natalia, however you want. Does the amount of washing reduce PFAS levels?
Yeah, I was gonna say, I mean, you could answer as well. So this is an interesting question. I mean, yes, I was gonna say yes, but not that I see that as a positive, because one of the main issues is like, when you're washing your gears, especially, I think like you have to take in consideration where you're washing it. So maybe if you're taking home and washing together with other clothing it, for example, together from the family, this could be also a way of, of exposure. So, but yeah, so because PFAS, they are both, as mentioned before, hydrophobic and hydrophilic. So because of their properties, they are also at some extent soluble in also water. But they can also be accumulating, and so that could be constituted also sort of exposure to other people, or it's going to end up also in the water cycle. So it's going to end up, so just think about that, you're washing it out and it's going it somewhere. So where is this? To the water treatment facilities, which then eventually they cannot really remove those contaminants, and then it can go all back to you, either, accumulating your food or, end up in the drinking water.
Great answer. And I would also point the audience to a paper published by Dr. Kenny Fent, where the chemical that they were looking at was PAHs, but he did find reduction by just using basic soap and water. So that doesn't mean it translates over to PFAS, obviously. But, as Dr. Suarez was saying, you just got to be sensitive about what you wash and where you wash it, because the fate and transport of that PFAS could environmentally affect other areas. So our next question from the audience says, great work. Were all the samples collected from used gear? If so, is the source of PFAS from the gear or a combination of gear PFAS at the fire scene? So I can take a stab at that one and then feel free to jump in, Dr. Suarez. So all the gear was sampled from one fire station in one day. And we worked with this fire station where they let us use gear that was in use, and we quickly sampled over a period of time there in the station. Because this was a pilot study and we just wanted to do proof of concept about, more about using the textile as a way of sampling, we did not survey the gear itself. So like, for example, how many years of service when it was purchased, and sort of the background information on the individual gear, nor did we get any information from the firefighters themselves, which is what Dr. Suarez alluded to, in that we are very interested now in collecting blood samples and trying to understand the linkage between serum PFAS and PFAS that's in the gear using this non-invasive method. So hopefully that answers the question. I don't know if there's anything else you want to add to that.
I think you did perfectly.
Okay. So we have a couple more questions. I think this one's maybe for you. In your opinion, are there certain types of PFAS species that are more significant for firefighter health? And if so, do you find these to be elevated in these types of studies? I would say, yeah, go ahead, you first.
Yeah, I mean, I was gonna say that, there are some types of PFAS. There are more commonly present in the foam. So, I mean, those should be probably the one they're going to be, having a higher exposure. I mean, the only thing that we have to take into consideration, as I mentioned before, is that some of those, in firefighting foams, like maybe we should have included some slide and showing you how is a normal composition of that, is that they can, some of them can actually degrade it into some of the perfluoroalkyl acids, like, such as PFOS and, PFOA. So I think like in the end, all of them are somehow significant for firefighter health. I was going to make, so at least a majority, but yeah, go ahead if you.
No, I was going to say, we're beginning to understand more within the firefighter context what health effects are seen from PFAS exposure. And so I think at a national level, we're at a point where we're trying to characterize what are those, the point sources of those exposures and what types of PFAS do they encounter. It can be very challenging. Sometimes some of the products that are produced with foams like the AFFF have proprietary blends of chemicals. And so we have to sort of backwards engineer and see what PFAS are in there to know those exposures. And so this project hopefully adds this new methodology of sampling in a non-invasive way that doesn't require destruction of gear, but still allows us to sample the gear over time and then be able to, further link it to health. So I think that's where we're at. Okay, our next question says, when will blood sampling take place? How many firefighters and what criteria will be set? Those are excellent questions for the next study. So traditionally, at least in Florida, the research work that's supported by the Firefighter Cancer Initiative is programmed a year in advance. So usually we start our programmatic year July 1st of every year. So in the next 6 months, if the state of Florida refunds us, we'll begin a new set of projects and continue some that are ongoing. And so we will initiate conversations again with Dr. Soares and the FIU team to see how we can start designing a study that would allow us to correlate both the sampling method of PFAS from the gear along with that in the human health. And I think we can also get potentially creative at looking at other ways of sampling in the fire station, not just the gear, but perhaps other spaces where we can assess PFAS transport and fate in the station. Okay, so the next question is, was PFAS found on internal layers of PPE? How many layers of fabric is in the PPE in the United States?
That's a great question. We actually haven't not look at an internal layer. So what we have been looking at is just swabbing at the surface of the gear. So we didn't look at PFAS, but there are a couple of studies, and I think Alberto, you mentioned it one, about looking at PFAS as the different. I mean, we know that the gears because our water repellent gears, they do have PFAS, and there was this study that it was mentioned here today that they did look at in these different layers, from the protective. I mean, from the gears. I mean, they didn't look at in the scuba, I guess, but they have found out that they have PFAS. So we are aware that the gears itself, they have PFAS, but our goal here was to actually try to sample the surface of the gears, and as a way, once the firefighters go to do the routine work, they can be, all of when they're using the foam, this can actually go into their gears as well, right? So they could be we just have to remember, injection might be one of the major pathways, for non-occupational personnel, but when we are dealing in case of here, that occupational work, there's a lot of other pathways inhalation that should be considered, for example, or so.
Yeah, absolutely. And I would just add that, in this particular pilot project, because we were trying to do this relatively quickly, only the outside of the gear was swabbed. So these are really great questions about sort of the different inner layers and the linings, particularly at the interface between, the firefighter skin and the turnout gear where sampling can take place. So I would just say, I think we are very excited about this method and being able to extract and collect and evaluate for PFAS. So now the future is open to how additional sampling areas can be done. It was very surprising to see that sort of the SCBA had lower levels also of it, because we think of the SCBA having contact, respiratory and inhalation. So I think this is a great first step.
And I was, just thinking about this question, it kept me wondering, I mean, one way that we could do it instead of looking at these different internal layers, I think ultimately we would be actually interested in assessing is there, by the use of PFAS, even if you were in the gears, could they still get pass all the internal layers and get exposed to the clothing itself? So that's probably also something very interesting to assess.
Yeah, to examine, indeed. Okay, so I think we answered the last two questions here. Are there any other questions that we could, or comments that we can answer from the audience? All right, terrific. So I'll turn it back over to Dr. Sally, who might have some closing remarks.
All right. Thank you guys for being here. Thank you so much, Dr. Gavan, Dr. Suarez, for presenting. I think this information was really important. And as you could see, many people are very interested in the topic. For everyone else, we hope to see you either in person or virtually next month at the 2023 International Firefighter Cancer Symposium. It will be hosted here in Doral, Florida on Thursday, February 23rd and Friday, February 24th. The 23rd will be at Fire Tower, local 1403, their union hall. And then the second day will be at MDFR's fire training. You can go to our website, you can register. Registration is free. And again, if you have any questions about attending the symposium, please feel free to reach out to myself, Tara Greenberg, or the FCI email address for any questions. But again, it is completely hybrid, so you can come in person one day, both days, or you can also just log in from your computer wherever you're at. But we are really excited. We have a great list of speakers coming from all over the world. We have several speakers that were scientists that were at the IARC monograph. So those who were sitting in the room making those decisions on how to classify firefighters' risk for cancer. And so that really will be the main topic of discussion throughout the day on Thursday. And then Friday, we have workshops, which we're really excited about. So we have one related to mental health in the fire service. We have the firefighter cancer prevention toolkit. We also have working with our firefighters in remediation of PFAS exposure. So we have 2 firefighters who are coming all the way from Australia to speak about this, which we're really excited about. And then we also have a full day of a scientific workshop. So everyone is invited to go to the different workshops, and we're really looking forward to it. If you have any questions, please always reach out to us. Another thing is on Saturday, the 25th of February, we will be having our Firefighters versus Cancer team for the Dolphins Challenge Cancer. If you are in the Miami area and would love to join, please reach out to us. We have been able to solidify donations to sponsor all firefighters and one family member to participate in the 5K or the bike ride. And so this covers all registration and fundraising fees. So please reach out to us. Please share the information with your departments and other colleagues. So we are excited for a very busy February. And then we will see you back in March, where we will be presenting the seminar series. Will actually be myself and Cynthia Beaver, who's one of our senior research associates on our cancer survivorship program in the fire service. So we're looking forward to that as well. Dr. Gavan, any other last minute announcements?
Nope, that was great. I just again want to thank Dr. Suarez and FIU for this collaborative research experience, which has been incredibly positive and shows, its dedication to the fire service. So thank you, Dr. Suarez.
Yeah, thank you so much. Yeah, it was a pleasure, this collaboration. And I was going to say, like, if anybody from the audience wanted to get in touch, I wanted to share my email, but I think I cannot do that.
We'll post it with this has been recorded for YouTube, so it'll be included, and they can
because I forgot to include it, I think, in my presentation. I normally do in the first slides, and then I completely forgot. I should have put in the last one. So if anybody wanted to get in touch, they didn't.
I was just able to add it to the chat so everyone can see it.
That's great then. Yeah, so if anybody wanted to get in touch or have any other questions about this about PFAS or measurement or anything related to that, you can get in touch with me as well.
Awesome. Thank you. All right, everybody, have a great afternoon. Be well.
Take care. Thank you.
Thank you.
Bye.
Bye.